Welcome back to the deep dive. Today, we are undertaking an intense and really necessary mission to map out the microscopic landscape of the accessory digestive organs.
Yep, we're talking about the liver, the gallbladder, and the pancreas.
And this system is, you could argue, one of the most complex in the human body. It's just this incredibly intertwined network of metabolic processing, storage, secretion, and hormonal control.
That's absolutely right. And for anyone studying this, this is our deep dive into Chapter 18 of Histology, a text and atlas. Our goal here is to, guide you step by step through this entire chapter.
So we're going to be extracting all the crucial details about the anatomy, the functions, the cellular mechanisms.
And the really vital clinical correlations that the text provides. You can think of this as the definitive shortcut to mastering the sheer volume of information this one chapter holds.
Okay, so let's start at the beginning. We'll set the stage with the largest internal organ and glandular mass we have, the liver.
It's a true biological behemoth. It weighs in at roughly 1,500 grams.
Wow. So that's about, what, 2.5% of your total adult body weight?
Exactly. Structurally, it's enclosed in this protective, dense, irregular connective tissue layer. It's called the glycine capsule.
And then most of that is covered by the visceral peritoneum on the outside.
Right. And the text notes the traditional anatomical divisions, the four major lobes you learn about. Right, left, quadrate, and caudate.
But those are mostly for, topographic description, right?
Yeah.
Just understanding where the liver sits relative to everything else.
Precisely. But if you're a surgeon or a pathologist, that anatomical division is actually less important than the functional division.
Ah, okay. And that distinction is critical.
It is. The functional segmentation of the liver isn't defined by the grooves on the surface. It's defined entirely by the distinct branching patterns of its blood supply and the bile drainage ducts that go along with it.
So a surgeon would need to know those functional segments to know which parts of the liver they can safely remove, making sure the blood supply and bile drainage stay intact for whatever's left.
Exactly. Before we zoom into the cells, let's just quickly touch on its origin, which is pretty surprising.
It starts developing super early, doesn't it?
Very early. As an endodermal evagination, which is basically just an outpouching from the wall of the embryonic foregut, specifically the part that becomes the duodenum.
And this initial structure, the hepatic reticulum, it gives rise to pretty much everything, right?
To all the hepatocytes, which form the functional part of the liver and the lining of the common bile duct.
And what I always find so neat is that another outgrowth from that same common bile duct, the cystic diverticulum, is what skins off to form the gallbladder and the cystic duct.
So they're all intrinsically linked. The liver, gallbladder, biliary system, they all share the single embryological lineage from the foregut.
All right, let's unpack the liver's function. If we had to summarize its job description, I'd call it the quadruple threat.
That's a good way to put it. It's an engine, a storage unit, a waste processor, and a regulator, all rolled into one.
So we can break its roles down into four major categories.
We can. First, it synthesizes and produces almost all circulating plasma proteins. Second, it's the primary storage and regulation hub for nutrients and key vitamins.
Third would be that relentless task of degrading drugs and toxins, the xenobiotics.
And fourth, it functions as both a crucial exocrine gland, producing bile, and an endocrine-like organ, modifying hormones from all over the body.
Let's start with production. The liver is a protein factory. It synthesizes most of our circulating plasma proteins, and the star of that show has to be albumin.
Oh, absolutely. Albumin is essential because it regulates plasma volume and maintains what we call colloid osmotic pressure.
Okay, let's break that down. What does that actually mean?
Basically, albumin acts like a water magnet inside your blood vessels. It's too big to easily pass through the vessel walls, so its concentration keeps water pulled into the circulation from the surrounding tissues. It maintains fluid balance and blood volume.
So if you don't have enough albumin, that pressure drops, fluid leaks out, and you get edema.
That's exactly it. And beyond albumin, the liver is central to managing lipoproteins. It makes and secretes VLDLs, very low-density lipoproteins.
And those are the main carriers for triglycerides, right, to get them out of the liver to other tissues.
Yes, for storage or energy use. It also produces all the major clotting factors.
This is a perfect time to look at folder 18.1 from the text, the clinical correlation on lipoproteins. It really helps visualize what these things are.
They're multi-component structures. Imagine a hydrophobic core of cholesterol and triglycerides all wrapped up in a hydrophilic shell of phospholipids and proteins.
which allows them to travel through the watery plasma.
And the textbook meticulously traces their origin inside the cell, which is a great example of how the hepatocyte organelles work together. The lipid part is made in the smooth ER, the SCR.
And the protein part, the apolipoproteins, they're synthesized in the rough ER, the RER.
Right, and they don't leave the cell until they're fully assembled and packaged, which all happens in the Golgi apparatus. Then the packaged VLDL is released into the space of Dc to enter the circulation.
We classify them into 4 classes based on density. Chylomicrons are the lightest. VLDLs carry triglycerides from the liver. Then there are LDLs.
And LDLs are the ones that carry cholesterol from the liver to peripheral cells. High levels of these are what clinically increase your cardiovascular risk. They're the bad cholesterol.
And the good guy.
That would be HDLs, high-density lipoproteins. Their job is the opposite, reverse cholesterol transport. They scavenge excess cholesterol from tissues and haul it back to the liver for processing or excretion.
So high HDL is a good thing. The liver is managing both synthesis and recycling.
And we should quickly mention the other key proteins, clotting factors like prothrombin and fibrinogen, which are vital for coagulation.
Plus all those carrier proteins, glycoproteins like haptoglobin and transferrin that transport metals and other compounds.
Exactly, so that's just production. Let's move to storage. The liver is a vital vitamin warehouse, especially for the fat-soluble ones, A, D, E, and K.
So vitamin A or retinol, the liver processes it, but the text specifically points out that it's stored not in the hepatocytes themselves.
But within the lipid droplets of the hepatic stellate cells, which we'll definitely get to later.
And when it's needed, it's released into the blood bound to retinol-binding protein. And a deficiency, of course, is famously linked to night blindness.
Right. Then there's vitamin D, cholecalciferol. The liver performs the first critical hydroxylation step, converting it into 25-hydroxycholecalciferol, the main circulating form.
And that happens before it goes to the kidneys for final activation. So without that liver step, the body can't use vitamin D, leading to things like rickets.
Then you have vitamin E, a powerful antioxidant. Its secretion is tied to VLDL assembly, which is interesting. Reduced levels are often seen in fatty liver disease, suggesting a breakdown in that pathway.
And lastly, vitamin K, indispensable for making those clotting factors. A deficiency leads to bleeding disorders.
The liver also manages complex metal homeostasis, especially for iron and copper. For iron, hepatocytes are major long-term storage sites, and the liver makes the proteins needed to recover iron from old red blood cells.
The body is really careful about iron because it's vital, but also potentially toxic.
Extremely careful, and copper regulation is even more delicate. Hepatocytes are the main site of uptake, and the key is regulating its excretion into bile and incorporating it into the transport protein, ceruloplasmin.
And this whole process is governed by a single enzyme, the copper ATPase or Wilson ATPase.
Which brings us right to a major clinical correlation, Wilson disease. This is a severe autosomal recessive disorder. It's caused by a mutation in the ATP7B gene, which codes for that Wilson ATPase.
So if that enzyme is impaired, copper can't be properly handled. It can't be put into ceruloplasmin or secreted into the bile.
And what happens then is a toxic cascade. Copper builds up to dangerous levels in the liver, causing severe cirrhosis. Eventually, it spills into the circulation and deposits in the brain and eyes, causing severe neurological damage.
This is a perfect illustration of how one single genetic failure in a hepatocyte can devastate the entire body.
Now, moving on to the liver's most famous role, detoxification, degrading drugs and toxins or xenobiotics.
And a lot of these are non-hydrophilic, meaning they're fat-soluble, so they can't just be excreted by the kidneys and urine.
So the hepatocytes have to convert them into water-soluble forms, a process that happens in 2 phases. Phase 1 is oxidation. This happens mainly in the SCR and mitochondria.
And this is where that huge family of cytochrome P450 enzymes comes in. They add polar groups like OH or Tokyo-OH groups to the molecule.
Right, which makes it a little more reactive and ready for phase 2. Phase 2 is conjugation.
This is where you add a big, bulky, water-soluble molecule like glucuronic acid or glycine to the substance.
And that addition ensures the final product is extremely water-soluble and can now be easily excreted by the kidneys or in the bile.
Okay, let's quickly summarize its role in the major metabolic pathways.
It's central to everything. Carbohydrate metabolism, storing glucose as glycogen, breaking it down to maintain blood sugar. Lipid metabolism, beta oxidation for energy, making cholesterol.
And protein metabolism, synthesizing proteins, deaminating amino acids, and making urea from toxic ammonia. It really does it all.
And now, the liver's explicit exocrine role, bile production. Bile isn't just waste. It's an essential secretion with bile salts that act like detergent to help absorb dietary fats.
Table 18.1 of the text outlines the composition. Mostly water, but the active ingredients are key.
Right. You have phospholipids and cholesterol, which are mostly recycled. You have the bile salts, which are the emulsifiers, and then the bile pigments, mainly bilirubin glucuronide.
And that's the detoxified end product of hemoglobin breakdown. It's carried to the gut for elimination, and importantly, it's the one major component that is not recycled back to the liver.
And finally, the liver's endocrine-like functions. It modifies hormones released from other places. For example, it converts the thyroid hormone T4 into its more active form, T3.
It also amplifies hormone action, like with growth hormone, GH, which is amplified by the liver-produced insulin-like growth factor 1, or IGF-1.
And the text gives a fascinating specific example of this.
The Bayaka pygmies of Central Africa.
Exactly. They have normal levels of growth hormone, but their genes mean they produce reduced levels of IGF-1. This failure to amplify the GH signal is what results in their short stature. It's a beautiful illustration of local liver regulation affecting systemic physiology.
And let's not forget, the liver is the main place where insulin and glucagon are broken down, cementing its role as the central metabolic regulator.
So now, moving from function to structure. We have to start with the liver's most defining feature, its dual blood supply.
It's totally unique, and it dictates, well, everything else about its structure and pathology.
The blood supply is literally a mixed bag. About 75% comes from the venous hepatic portal vein. This is blood from the gut, so it's nutrient-rich, toxin-rich, but oxygen-poor.
And the other 25% comes from the arterial hepatic artery, supplying the high-oxygen blood.
Both of these vessels, plus the common bile duct and lymphatics, enter the liver at the hilum, or the porta hepatis. And here's the key flow distinction to remember.
The mixed blood flows in towards the center of the lobule, while the bile flows out towards the porta hepatis.
In the exact opposite direction, centrifugal flow for bile.
And these three tubes, the branch of the hepatic artery, the branch of the portal vein, and the bile duct, they travel together in connective tissue tracts,
which we call portal canals. And that trio is the famous portal triad.
Although the text calls the term portal triad a bit of a misnomer.
Right, because lymphatic vessels are always there too, making it more of a portal tetrad if you want to be precise.
Okay, let's trace that flow microscopically. The mixed blood enters the sinusoidal capillaries, or sinusoids.
It percolates across the plates of hepatocytes, allowing for all that metabolic exchange. Then the blood drains into the collecting channel at the center, the terminal hepatic venule, or central vein.
And from there, it flows out through sublobular veins and eventually exits the liver via the hepatic veins to the inferior vena cava.
So the four main structural components are the parenchyma, which are the hepatocyte plates, the connective tissue stroma, the sinusoidal papillaries, and the persinusoidal spaces, or spaces of Dissy.
To really understand the liver, you have to grapple with the three different functional models of its organization. They all describe the same tissue, but prioritize different functions.
First up is the classic hepatic lobule. This is the traditional hexagonal unit, and it's centered entirely on blood flow.
So the central vein is in the middle, and the portal triads define the six corners of the hexagon.
And the textbook points out that while this shape is really clear in some animals like pigs, because they have a lot of connective tissue separating the lobules,
in humans, that connective tissue is minimal. So we basically have to draw imaginary lines between the portal canals to see the hexagon.
Exactly. The second model is the portal lobule. This one focuses on the exocrine function bile secretion.
So it's roughly triangular in shape. Its axis is the bile duct in the portal triad, and the three corners are defined by the three nearest central veins.
It's a purely physiological concept. And third, the model that's most relevant for pathology and metabolism, the liver acinus.
This one is described as a lozenge or diamond shape. It's the smallest functional unit. Its short axis is defined by the branches of the portal triad, and its long axis runs between two central veins.
And the acinus model is so important because it lets us visualize zonation. This results directly from the oxygen and nutrient gradient in the blood flow.
A gradient that causes cells in different regions to have different metabolic properties.
The acinus is divided into three concentric zones, which are shown really clearly in figure 18.6b. Zone 1 is closest to the arterial and portal blood supply. It gets the highest concentration of oxygen, nutrients, and critically toxins.
So because it gets the freshest blood, Zone 1 is the last zone to die if circulation is impaired, and it's the first to regenerate.
But conversely, it's the first zone to show damage, like bile stasis, if the bile duct is blocked because it's at the start of the biliary channel.
And Zone 3.
Zone 3 is the opposite. It's the farthest from the blood supply, right next to the central vein. It gets the most oxygen-depleted and nutrient-depleted blood.
So Zone 3 is the first to show signs of ischemic necrosis cell death from lack of oxygen. It's also the first to accumulate fat.
And it's generally the last to respond to toxic substances and bile stasis. And Zone 2 is just the intermediate zone between them.
This zonation isn't just theoretical. It really defines pathology, as we see in Folder 18.2 congestive heart failure, acetaminophen overdose, and liver necrosis.
Right. In cases of hypoxia from something like congestive heart failure, the whole body suffers from low oxygen. But because Zone 3 gets the poorest oxygen supply to begin with, it's the first to die.
And that leads to ischemic necrosis centered around the central vein, which is called centrilobular necrosis.
It's purely an oxygen gradient effect. And the exact same pattern is the hallmark of acetaminophen overdose, which is the leading cause of acute liver failure in the U.S.
Why Zone 3 for that?
When you take high doses of acetaminophen, it's processed by the cytochrome P450 system into a toxic intermediate called NPQI. Normally, NAPQI is quickly detoxified by the liver's glutathione.
But a high dose just completely depletes that glutathione.
And the toxic, unconjugated NAPQI that's left binds to hepatocyte organelles and kills them. Zone 3 is hit hardest, maybe because P450 activity is highest there, or it's just the most metabolically stressed zone.
The result is rapid, massive cell death right around the central veins.
Okay, zooming in now to the microscopic level. Let's look at the hepatic sinusoids. These are the low-pressure vascular channels where all the exchange happens.
And the lining of these sinusoids is really unique, right? It's key to how the liver works.
It is. It's a thin, discontinuous endothelium. This means the cells have these large fenestrae or holes that don't have diaphragms, and there are big gaps between the cells.
So it's like a sieve or a colander.
That's a great analogy. This discontinuous nature is crucial because it lets plasma flow freely out of the vessel and into the space around the hepatocytes for a maximum exchange.
And living right inside this sinusoidal lining are the specialized immune cells, the stellate sinusoidal macrophages, which we all know as Kupffer cells.
The text clarifies they're a mixed population. Some are self-sustaining residents from the yolk sac. Others are monocytes from the bone marrow that come in after injury.
Functionally, they are incredibly phagocytic. They're the liver's primary defense, acting as antigen-presenting cells to fight anything coming from the intestines.
And they clear out old, damaged red blood cells, recycling something like 90 to 95% of the body's iron supply.
Let's get into that iron recovery mechanism. A Kupffer cell eats an old red blood cell. What does it do with the iron?
It has to store it safely. It sequesters the iron inside a big protein complex called ferritin, which can hold up to 4,500 iron ions.
And then it carefully exports that iron back into the circulation using a protein called ferroportin-1.
Where it binds to transferrin for safe transport. Now, if the Kupffer cell has too much iron, the ferritin aggregates into insoluble deposits you can see under a microscope.
And those are hemosiderin granules.
And that's the clinical connection. Too much hemosiderin in parenchymal cells is hemochromatosis. In macrophages, it's hemosiderosis. And since iron is paramagnetic, you can actually quantify the overload non-invasively using an MRI.
Moving just past the endothelial lining, we get to that crucial exchange zone, the parasinusoidal space, or the space of Disse.
This microscopic gap is between the discontinuous endothelium and the plasma membrane of the hepatocytes. This is where everything happens.
Nutrients and plasma flow in, and things that hepatocyte makes, like plasma proteins, flow out.
And the surface area here is massive because the hepatocytes extend these long microvilli into the space, increasing the exchange surface by up to 6 times.
Also tucked away in the space of Disse are the hepatic stellate cells, which used to be called Edo cells.
Under normal, healthy conditions, their job is pretty simple. They are the main storage site for vitamin A, which they keep in little cytoplasmic lipid droplets.
But these are the cells that can cause severe pathology.
They are. In chronic inflammation or injury, the stellate cells undergo this dramatic transformation. They lose their vitamin A and differentiate into highly active myofibroblasts.
And that's bad, because these myofibroblasts are the engine of fibrosis. They start pumping out huge amounts of type I and type 3 collagen.
And that collagen deposition creates scars, disrupts the whole architecture, and leads directly to cirrhosis.
On top of that, they become contractile. They develop alpha-smooth muscle actin, which lets them squeeze the sinusoids.
And that increases vascular resistance, which is a major contributor to the deadly condition known as portal hypertension.
Okay, let's quickly trace the lymphatic pathway. The liver makes a huge amount of lymph.
Right, the plasma that filters into the space of Dissy eventually drains into the periportal space, or space of Moll, which is right between the stroma of the portal canal and the outermost hepatocytes.
And from there, it enters lymphatic capillaries that run alongside the portal triad. It's a huge volume. 80% of the lymph that enters the thoracic duct actually comes from the liver.
And finally, the cells themselves, the hepatocytes. These are large polygonal cells, making up about 80% of the liver cell population. They're incredibly robust, with a 5-month lifespan and an amazing capacity for regeneration.
Microscopically, they're often binucleate, and interestingly, most adult hepatocytes are tetraploid.
Meaning they have 4 sets of chromosomes. This high degree of ploidy likely contributes to their huge metabolic capacity and their ability to regenerate so well.
Looking at their surfaces, there are 3 distinct domains, as shown in figure 18.13. You have the surface facing the parasinusoidal space, the lateral surface facing other hepatocytes, and the apical surface facing the bile canaliculus.
And their cytoplasm is just a testament to their synthetic activity. Strong basophilic regions from the RER, a massive 800 to 1,000 mitochondria per cell.
Multiple Golgi complexes, lipid droplets, and lipofuscin pigment, which is just leftover lysosomal gunk, a sign of the cell's long, hard-working life.
Let's focus on 2 key organelles. First, peroxisomes. Hepatocytes have up to 300 of them. They're crucial for lipid metabolism and breaking down fatty acids.
And they produce hydrogen peroxide as a byproduct, which is immediately degraded by the catalase enzyme inside them.
And a surprising fact from the text, about half of all ingested ethanol is actually converted to acetaldehyde by enzymes in these peroxisomes, which is a detoxification role separate from the P450 system.
Okay, and second, the smooth endoplasmic reticulum, the SER.
This is extensive and highly dynamic. It has all the enzymes for detox, conjugation, cholesterol synthesis, and making the lipid part of lipoproteins.
And the SER shows incredible plasticity. When you challenge the body with large amounts of alcohol or certain drugs, the SER undergoes massive hypertrophy. It actually grows larger.
And while this increases the liver's detox capacity, the text notes a paradoxical risk. The increased P450 activity can actually make some compounds more toxic initially, magnifying the damage. A classic example is the increased toxicity of carbon tetrachloride when the SER is hypertrophied.
All right, since hepatocytes are constantly making bile, we need to trace where that fluid goes. That brings us to the biliary tree.
This is an intricate 3-dimensional system of increasing channels that transports and subtly modifies bile on its way to the gut.
And the entire tree is lined by these epithelial cells called cholangiocytes.
Right. They're cuboidal to columnar, depending on the duct size, and they have a primary cilium on their apical surface that's thought to sense bile flow.
The starting point is the bile canaliculus. This isn't really a duct, is it?
Not in the conventional sense. It's just a tiny channel formed by deep grooves in the apical surfaces of 2 adjacent hepatocytes, sealed off by tight junctions.
And the flow is centrifugal, moving away from the central vein toward the portal triad.
The canaliculi then drain into the first true duct-like structure, the canal of Herring.
This is a short channel with a mixed lining, partly hepatocytes, partly cuboidal cholangiocytes.
And clinically, the canal of Herring is indispensable because it harbors the liver progenitor cells, the hepatic stem cell reservoir.
So after massive liver damage, these are the cells that proliferate and can differentiate into new hepatocytes, or bile duct cells, providing a backup repair mechanism.
From there, bile enters the intrahepatic bile ductules, then the interlobular bile ducts of the portal triad, which merge to form the right and left hepatic ducts.
And beyond the liver, the extrahepatic bile ducts begin. The right and left ducts merge to form the common hepatic duct,
which is then joined by the cystic duct coming from the gallbladder to form the common bile duct.
That cystic duct has those spiral folds, the spiral valve of Heister, and it's mostly a passive conduit for bile moving in or out of the gallbladder.
Right. The common bile duct extends down to the duodenum, ending at the hepatopancreatic ampulla, or ampulla of Vater. And its release is controlled by the sphincter of the hepatopancreatic ampulla, or sphincter of Oddi.
And when the sphincter of Oddi contracts, bile can't get into the duodenum, so it backs up the cystic duct and flows into the gallbladder for storage and concentration.
So now, for that storage unit, the gallbladder, this pear-shaped sac can hold about 50 milliliters of bile. Its entire highly specialized function is storage and concentration.
And it's incredibly effective. capable of removing about 90% of the water from the incoming bile.
which dramatically increases the concentration of bile salts, pigments, and cholesterol up to tenfold. Contraction is primarily stimulated by the hormone CCK in response to fat in the intestine.
Looking at its histology, the mucosa is deeply folded when it's empty. The epithelium is a simple columnar layer of very tall cholangiocytes.
And histologically, these cells look a lot like intestinal absorptive cells. They have short microvilli, robust junctional complexes, and a ton of mitochondria to fuel their active transport work.
The gallbladder wall is also unique. The text points out that it's different from the rest of the GI tract.
Very different. It completely lacks both a muscularis mucosa and a submucosa.
So external to the lamina propria is the thick muscularis externa, with randomly oriented bundles of smooth muscle. Their contraction is what forces the concentrated bile out.
And histologically, you can sometimes see these structures called Rokitansky-Aschoff sinuses. These are deep, crypt-like outpouchings of the mucosa that extend all the way through the muscularis externa. They're often seen as a risk factor for gallstone formation.
Okay, the critical function of bile concentration relies on this active, coupled transport of salt and water across the epithelial cells.
It's an elegant mechanism. Step 1. The cells actively pump electrolytes, sodium, chloride, bicarbonate out of the cell and into the narrow lateral intercellular space. This is fueled by AT passes on the lateral membrane.
Step 2. This massive influx of salt creates a steep osmotic gradient in that narrow space,
which then forces water to follow the salt passively. This water movement happens through specialized water channels, aquaporins, AQP1 and AQP8.
Step 3. The influx of water swells the space, creating hydrostatic pressure. This pressure forces the now isotonic fluid out of the space and into the fenestrated capillaries of the lamina propria where it's carried away. And this just happens continuously.
Okay, we now transition to the final major accessory organ, the pancreas. This elongated gland sits with its head cradled by the duodenum and its tail extending toward the spleen.
The main pancreatic duct of Wirsum runs the length of the gland and joins the common bile duct at the ampulla of Vater.
And unlike the liver, where the functions are mixed within the same cell, the pancreas has structurally distinct components for its dual function.
You have the exocrine component, which makes digestive enzymes, and the endocrine components, the islets of Langerhans, which secrete hormones like insulin and glucagon.
Focusing on the exocrine pancreas first. It's a pure serous gland organized into acinar units formed by pyramidal serous cells.
And the most unique and diagnostic feature of the pancreatic acinus is the centroacinar cells.
Right. These are the pale staining squamous cells that form the very beginning of the intercalated duct inside the acinus itself. It's like a straw pushed into a cluster of berries.
And the surrounding acinar cells are classic protein factories. Strong basophilia at the base due to tons of RER, and large acidophilic zymogen granules packed in the apical cytoplasm.
These granules contain a full suite of inactive digestive enzyme precursors, proteolytic enzymes like trypsinogen, amylolytic enzymes like alpha amylase, lipases, and nucleolytic enzymes.
The inactivity part is paramount. They have to stay inactive as zymogens until they reach the small intestine to prevent the pancreas from digesting itself.
Exactly. The duct system starts with those centroacinar cells, which are continuous with short intercalated ducts. These drain into intralobular collecting ducts. And a key point, the pancreas has no striated ducts.
The acini secrete a small volume of protein-rich fluid, but the intercalated duct cells are responsible for secreting a huge volume up to 1 liter a day that is uniquely rich in sodium and bicarbonate.
And that bicarbonate is absolutely essential for neutralizing the acidic chyme coming from the stomach, creating the perfect alkaline environment for the digestive enzymes to work.
This secretion is under tight hormonal control. The hormone secretin stimulates the duct cells to secrete that bicarbonate-rich fluid.
And CCK, cholecystokinin, stimulates the acinar cells to release the zymogen proenzymes.
Now for the final segment, the endocrine regulator, the islets of Langerhans. These are discrete clusters of cells scattered throughout the pancreatic tissue, most numerous in the tail.
They're only about 1 to 2% of the pancreatic volume. In H&E sections, they just look like lighter, paler staining clusters. And they're highly vascularized with fenestrated capillaries for rapid hormone release.
While they look uniform in H&E, special techniques let us define the 3 principal cell types.
The majority are the B cells, or beta cells, making up 60 to 70% of the islet. They're centrally located and secrete insulin.
Next are the A cells, or alpha cells, about 15 to 20%. They're usually located on the periphery and secrete glucagon.
And finally, the D cells, or delta cells, 5 to 10%, also peripheral, and they secrete somatostatin.
So let's talk functions. Insulin is the primary hormone for glucose regulation. Its job is to decrease blood glucose.
It does this by stimulating several things at once, upregulating GLUT4 transporters to increase glucose uptake, promoting storage as glycogen, encouraging its use through glycolysis, increasing lipogenesis, and promoting protein synthesis.
And an insulin deficiency, of course, defines diabetes mellitus. Folder 18.3 even links insulin resistance to Alzheimer's disease, suggesting it may contribute to neural degeneration.
Right, and conversely, glucagon does the opposite. Its job is to increase blood glucose by stimulating gluconeogenesis and glycogenolysis in the liver.
And somatostatin acts as a local regulator, inhibiting the secretion of both insulin and glucagon, sort of calming down the local interconnectivity.
Now, for a quick deep dive into folder 18.4 insulin synthesis, it's a brilliant example of post-translational processing.
It starts in the RER as a single polypeptide chain called preproinsulin.
The signal sequence is immediately removed, resulting in the shorter chain, proinsulin.
Then in the Golgi, proinsulin is cleaved, removing the C-peptide, or connecting peptide. What's left is the active insulin molecule, the A chain and B chain, held together by disulfide bridges.
And that C-peptide is clinically important. It has no known function, but it's secreted in equal amounts with insulin and has a much longer half-life.
So measuring C-peptide levels is the best way for doctors to assess a patient's own B cell function, which is critical for classifying diabetes or monitoring transplants.
Finally, let's wrap up with the remarkable regulation and blood supply of the islets. Insulin release is stimulated by high glucose. Glucagon release is stimulated by low glucose.
And this leads to one of the most fascinating structural relationships in the whole system, the cascading perfusion. The islets are tiny, but they get a disproportionate 10 to 15% of the total pancreatic blood flow.
The blood flow is sequential. Capillaries often perfuse the peripheral A and D cells first, before reaching the central B cells. But here's the critical part. The efferent capillaries leaving the islets then branch out to supply the surrounding exocrine achini.
Wow. So the hormones from the islets don't just go into the systemic circulation, they're delivered locally and immediately to the exocrine tissue.
Exactly. It ensures that the body's metabolic state, regulated by the endocrine output, instantly controls the digestive enzyme output required from the exocrine pancreas.
There was a tremendous journey through the histology and physiology of these three organs. The integration is really seamless.
It truly is.
Okay, let's quickly recap the essential takeaways. The liver is the ultimate multitasker, defined by its dual blood supply and its zonation, which determines its metabolic activity and its vulnerability.
Right. Remember that critical distinction between zone 1 for survival and regeneration and zone 3 for necrosis and fat accumulation?
The gallbladder is a master concentrator, relying on its unique lack of structural layers and its sophisticated active salt transport, coupled with passive water movement.
And the pancreas manages its dual roles through separate structures. The exocrine tissue, uniquely defined by those bicarbonate-secreting center acinar cells, and the endocrine islets that so tightly regulate glucose.
And we just covered that final crucial point, the cascading blood flow, where capillaries from the islets directly supply the exocrine tissue, instantly coupling metabolic demands with digestive output.
And this functional connection leads us to our final thought. If the metabolic state, regulated by hormones, instantly controls digestive output via this local delivery system, how might a chronic condition like type 2 diabetes, which involves long-term insulin resistance and altered hormone levels, eventually lead to structural and functional deterioration, maybe even fibrosis, within the surrounding exocrine pancreas itself?
It's a compelling question. It really shows how deeply interconnected these systems are, from the microscopic cell surface all the way to systemic health. Thank you for joining us for this incredibly detailed deep dive into the histology of the liver, gallbladder, and pancreas.
We'll catch you on the next one.